Yeah.
You understand in German?
Yeah, it's German. My German is pretty good. So, no, kidding. Hello, everybody. Or in the language of Bremen, I think it's Moin or Ahoy. Yeah. So, this is Bilal. I am a master's student at the Institute of Biomedical Engineering at Boğaziçi University. And today I will talk about the preclinical quantitative MRI research that we carry out at the Multimodal Imaging and Physiology Lab, aka MIMLAB. So first I'll talk about MIMLAB and TACTIX 1004. I'll talk about what multiparametric quantitative MRI is and why we're interested in it. And I'll give some examples of how we use it at MIMLAB. And then I'll wrap up with some limitations and future directions. Since we're a young lab, we don't have many collaborations. So I wanted to use this opportunity to introduce us and talk about research. Yeah, this is MIMLAB. It's a young lab established in 2022 by our PI, Bernard Özbay. You can probably see some familiar faces here in the hall, but it was winter, so everybody was trying to hide in their jackets. So yeah, we have many members. Our PI is Bernard Özbay, as I said. We have 1 postdoc, 4 PhD students, and 8 master's students. We already have 1 master's graduate officially in the last year, and hopefully 3 this year. Yeah, including me, hopefully. Fingers crossed. So we'll see. And yeah, we've had many interns come in our lab. We have many projects, ongoing projects now on subjects like cognition, memory, attention, mental stress, caffeine, and many others. But today I will talk about this one, which is about aging in animals. You can find further information in our website. So, TACTIX 1004, what's that? So, TACTIX 1004 is generally a support program given by TACTIX. It's quite a prestigious one. It's given for high impact research in strategic areas like nanotechnology and neurotechnology. And one of the recipients of this support program in Turkey is the Neurotechnological Solutions Platform, led by Professor Can Yücesoy at our institute. As you can see, it has many sub-projects like P1A, P1B, and until P14. It has many partners from both industry and academia, so 13 universities and 10 industry partners. And you can see here some of the examples of the partners, Boğaziçi University, Bilkent, and some other companies. Examples of the sub-projects are for example, the P13 project is led by Professor Gülcan Erçetin, and the P11 is led by Professor Esin Öztürkçü. She's among us here. And it's on application and innovation platform for small animal imaging to study aging. And a sub-project of that is what I am working on with Assistant Professor Bernard Özbay, which is namely identifying biomarkers characterizing aging through multiparametric MRI in animal studies. So multiparametric MRI, I've been talking about this. I've said the word probably too many times. So yeah, you can find more information on the website, but unfortunately this is in Turkish, so yeah. So what is multiparametric quantitative MRI? So yesterday, Professor Matthias gave a great overview of MR physics. That made it very easy for me for today. So generally in MRI, protons in water molecules are targeted because they're abundant in the human body. Usually they're spinning in random directions, so the magnetic fields that they generate are canceled out. So we're not magnetic beings, fortunately. But when we apply an external magnetic field, all these spins are aligned along or against the magnetic field. And then they spin in that direction. When we further apply an RF pulse, which we grappled with yesterday a lot using the MR education tool, they tip away from the axis of the external magnetic field. And then when we turn off that RF pulse, they start the process of relaxation. And relaxation has many components. It has a transverse and a longitudinal component, as you can see also in this one. So here, these are different parameters. As I said, different components of the relaxation process, a T1 and a T2 process. They look a little bit different, but depending on the time that you choose, the same tissue can look different, really. So you can see that here a particular voxel may look dark here, but then bright here, because that depends on the time that you choose to acquire the image. And that's why this kind of images are called qualitative images, or T1, for example, weighted, or T2 weighted images. These are qualitative, like they're subjective. They depend on the time that you choose. They depend on the scanner that you're using, like the values themselves. But each kind of tissue has a very characteristic curve. So these are exponential curves. And to characterize these curves, since these are exponentials, what characterizes exponential curves are the exponents. And the exponents are actually the T1 and T2. These are signal models. You can derive the models from the Bloch equations. But that parameter is very characteristic to each tissue, and it doesn't change because it's an inherent property in the tissue. I can give a simpler example of that. Imagine that you're taking the picture of a tree. If you go closer to the tree, the tree will look bigger or larger in the picture. If you move further from it, away from it, it will look smaller in the picture. But that doesn't mean that the length of the tree changes. It's actually your distance. It depends on the distance that you choose to take the picture. But maybe you have a magical device and you can extract the length of the tree using the qualitative images that you acquired. And that's what quantitative MRI is. Quantitative MRI is when you use these qualitative images, and using signal models, you can get the quantitative. You can get actually the characteristic T1, for example, time for a specific tissue or voxel. I hope I explained that well. As I said, T1 and T2 are different parameters, but there are also different other parameters. We have susceptibility, we have T2 star relaxation time. So why use multiple parameters? Why not use one? Why not use only T1 or T2? So the thing is, the brain has tissue properties like myelination, water fraction, iron deposition, and brain structure. And these properties, they all influence the imaging parameters that we're acquiring, like the T1 time, the T2 time. Something is happening here, okay? Or the susceptibility. QSM stands for quantitative susceptibility mapping. So, or the R2 star relaxation rate. So these properties actually affect these parameters, and they affect them in different ways. So water content affects T1 values, but maybe not susceptibility values. And similarly, iron deposition may impact QSM or susceptibility, but not necessarily T1. So when you use all of them, they complement each other. You get a more comprehensive understanding of what you're looking at. How do we do that at MIMLAB? The TACTIX 1004 project that we're working in is an aging project. So far, we've had 18 young female Western rats scanned using a 7 Tesla preclinical MRI. It's an impressive one. It's the first one in Turkey. And it's in our campus in the LifeSci Targeted Treatment Technologies Animal Imaging Unit. And Dr. Uluc is also among us here. After acquiring, we acquire multiple images. We acquire T1 maps, T2 maps, multigradient echoes for QSM and R2 star, and anatomicals, or T1 weighted and T2 weighted. And then we do some processing, and then we get quantitative MRI. For example, after fitting the T1 maps and extracting the T1 relaxation times from each voxel, we can find the average T1 times in each region of interest, like in the gray matter, in the white matter, ventricles, and so on and so forth. Here below, you can see an example slice from a rat brain. I don't know if you've ever seen a rat brain, but imagine that the rat is lying like that. Their spine is looking upward, and now we're looking at their brain from this direction or from this direction, okay? And it's called a coronal slice. Similarly, also, we can get the T2 values, the average T2 values for each region of interest. And you can also see some example slices from a T2 map, a quantitative one. For QSM, it's a bit more complicated. We have to go through multiple pre-processing steps. Since I said multi-gradient echo, so it has multiple echoes, we need to combine these echoes. And in literature, there are many ways. We prefer the weighted phase increment. And then you can see here that there are phase discontinuities, and this makes it difficult for us. So we apply phase unwrapping, too much detail, like pre-processing detail, but after that we apply background field removal using V#, and then we apply QSM star for dipole inversion to finally get the susceptibility values for each, like for the whole brain. And then we can do some ROI-based analysis or region of interest-based analysis. Here you can see examples like average susceptibility values in units of parts per million. This is a quantitative image for each region of interest. And you can see here example slices from the rat brain, like from a susceptibility map. This time we're looking at a different direction. So if the same animal is lying like this, we're looking from upward now. And you can see here a triangle-like shaped structure. This is a deep brain structure. And you can see that it's already brighter than its surrounding. And these structures are known for iron deposition with aging. So as the animal ages, more iron gets deposited there. So you can already see that it's visible. For R2 star, we also applied signal fitting or modeling. And we got these average values for each region of interest. And also see here some example images. We're looking from the same direction again, up from above the animal, the subject. But you can see a systematic outlier. 3 subjects are systematically higher than all other subjects. And when we further investigated this, we found out that these values do not belong to the oldest. So you can see here that there are oldest subjects, but they belong to the least heavy animals. And this kind of suggested that it has to do with the tissue content. Since they're less heavy, maybe the tissue content is different. These results were presented in ESMRM 2024 and ISMR 2025. Lean, my teammate, used a UNet, this is another example, he used a UNet model to extract the brain. She sliced the 3D volumes into 2D slices to make the data more for training and testing and validation. She did some data augmentation, normalization, and resizing, and she got pretty impressive matrices. So a dice coefficient of 97%, high sensitivity, Tversky, and Jacquard index. You can also here see visible comparison between the different methods. So FSL sometimes cannot detect, like here, cannot detect brain tissue, but the UNet detected it very well. Not only that, but we also get, it's generalizable to other kinds of images. So Lean used T1 weighted images, but the model generalized well for T2 weighted images and for multi-gradient echo images. And you can see that how FSL actually fails in some examples here, whereas the UNet works well. These were also presented in ISMRM 2025. Then she used this UNet model to extract the brain and then calculate somehow, after doing registration and segmentation, to calculate the volume of each kind of tissue, like the white matter or the gray matter, or some particular regions of interest, like the thalamus, for example. And then this provides a baseline for future longitudinal works. It's an aging project. And this was submitted to ASMRMB 2025. You can see here examples of some segmentations of tissue, like here, the corpus callosum and other kinds of tissue. I'm almost there. Throughout our research, scans, we came across an interesting case of cortical malformation. You can see here a T2 weighted image of the abnormal cortical malformation case versus a healthy control. There's clearly something wrong here. So a flare was acquired, fluid attenuated inversion recovery. It was for a 71 days old subject, and we can still see the malformation here. Then we applied a multiparametric approach again. We got the T1 map, the T2 map, the R2 star, T2 star, and QSM, and then checked the values from that malformation region. And what we found out that T1, T2, and T2 star times have increased, which suggests an increased water content and a reduced tissue complexity in myelination. Whereas on the other hand, R2 star rates and QSM or susceptibility decreased, and this suggested the presence of CSF-like fluid and altered microstructure. And as we expected, values from the unaffected brain regions were consistent with age-matched healthy controls. This was also submitted to ASMRMB 2025. So, this is advantageous. Multiparametric approach is advantageous. This is an ongoing work. We're acquiring more data points to understand the aging process and how it affects the brain and how it affects different parameters. However, this comes with some trade-offs. Since we use multiple parameters, we need to acquire many images, but we cannot keep the subject inside the scanner forever, and we cannot use the scanner forever because it heats up, so we cannot use it forever. So we have to make some trade-offs in terms of resolution, but then lower resolution actually affects the accuracy of segmentation and ROI-based analysis, especially that the structures in the rat brain are small. So we need to some good resolution. So yeah, in the future, optimization of sequences can enable more sophisticated scans with higher resolutions. Yeah, huge thanks to the team, MIMLAB and computational imaging team. They're here, and for the imaging support by Dr. Uluç and his teammates. Thank you so much for listening. I can take questions. Any questions? Yeah.
Do you optimize your sequences yourself, or do you have your own protocols, or is it something that comes with a scanner, for example, in a selection of, I don't know, click box, brain, quantitative, whatever?
Okay, so the question is, do we optimize the sequences ourselves, or does the scanner software has options for that? And the answer is no, we don't do it ourselves, and the scanner software doesn't have it, so it's neither. But yeah, maybe it's something to work on in the future, adapt to the Gamma Star software somehow. So maybe it's
when you do your scans, so how do you choose a protocol? Maybe.
Oh, of course, the scanner has some prepared protocols, multi-gradient echoes, multiple sites, multiple echo, IR flash, yeah.
So you take these and then...
Yeah, change the TE, TRs, but that's on the side of the imaging team. It's beyond my expertise.
Okay.
Yeah. Any other questions?
Have you tried looking at the aging markers? Did you get to collect longitudinal data?
Not yet, but we've had preliminary data from before the project started, and we realized there was a change in T1 and T2 values as the rat aged, but it was not a longitudinal work, so it was from different subjects at different ages, and we realized the decrease in T1 or T2, for example. But
did you match to any behavioral?
No, because it wasn't acquired by us. It wasn't within the scope of the project. It was preliminary data. We shared that. We presented that in, I think, ESMRM 2024. Anything else? Yeah.
I don't know if it's it's kind of you showed this graph of 2 outliers, I think. Was it 2?
Yeah, 3.
3, okay. And you said it's related to their lower weight. I was actually wondering, of course, you would need much bigger cohort for that, but there are studies showing that caloric restriction, for example, is related to longevity, and maybe somehow these animals are eating less, and they weigh less, and that's actually related to not the tissue contact, but like they are aging less maybe,
Mm-hmm.
or you know like rather like they are seeing the effects of aging. Yeah, basically they are slower in aging. So did you think about that and maybe trying to, yeah, these are of course outliers, but maybe you could separate the group into like higher weighing or lower weighing animals and yeah, relate that to the aging parameters as well.
I don't think that was considered, but yeah, maybe what you're saying is.
Yeah, I don't
That
know. Of
makes
course,
sense.
you would need to observe the behavior of animals if they are eating less or yeah.
Yeah, could be. Yeah. Thanks. Any other comments or questions? No? Okay then, I think end it here. Thank you so much.